computational biology. There are different definitions, one for computational biology here, one for bioinformatics. All of the talks, by the way, will be on our website. Some of you already know that. Ultimately, whether you talk about computational biology or bioinformatics depends on which side of the Atlantic you're sitting on. In Europe, people talk about bioinformatics, about what I do here. In the US, typically it's referred to as computational biology, while bioinformatics is there meant to be more anything that has to do with maintaining databases. Who are we? So my name is Burkhard Rost. I'm currently professor for computational biology and bioinformatics in the informatics department here. I'm a fellow at the Institute of Advanced Studies at the TUM. I'm an affiliate professor in Weinstefan, Freising. I'm an affiliate professor at Columbia University at the College of Physicians and Surgeons in the Department of Biochemistry and Molecular Biophysics. My background is I studied complex systems. I did a physics master in Gießen University in Germany, then went on to Heidelberg, did my master itself on neural networks, studied the complexity of sensor networks in Heidelberg, Bochum, Princeton, Harvard, and UCSF, did a PhD and a postdoc in computational biology at the MBL in Heidelberg, did a brief study at the European Bioinformatics Institute that was started then, the EBI at Cambridge, UK, went back to the MBL, went to a company, Lion Biosciences, and then became professor at Columbia University. In 2009, I went to Germany as an Alexander von Humboldt professor and have been here ever since. 2006 to 2014, I was the president of the International Society for Computational Biology, which represents this field. Clearly, for me, my passion is my job. It's a lot of fun. It is not so much money. There's a lot of travel. It's friends. What the movie here shows is just 1.5 years when I sort of gave talks on 5 continents. Actually, last year I did the same, even 6. I've given now almost 200 invited talks in 32 countries, 255 publications, and I co-published with 929 scientists, or more than that. The work has been cited over 34,000 times with an H-index of 84. Here's the group at 2 different time points at meetings here, at a meeting in Vienna, at a meeting in Berlin, and here in Garching. That is sort of the stem of the current group, shrinking. When you want to write an email to me, don't use any address. You find tons of my addresses on the internet. They all don't work, because essentially email doesn't work with me. I can't handle email. If you want to send an email, do it please to Inga, assistant at Rost Lab. If there are specific questions, Lothar is responsible for the teaching. And there's a teaching at rostlab.org address for that. Today, I'm going to briefly introduce biology, tell you a little bit about what organisms are, what genes are, what the central dogma of molecular biology, or what often is considered the central dogma of molecular biology, introduce proteins, and in the next lectures we go a little bit deeper into this, and then begin to talk really about protein structures and the way we can predict them. There is no Nobel Prize for informatics, as you all know. Here's a question mark, as if there would be an opportunity. In fact, there is something, in some sense, like it. In 2013, the Nobel Prize in Chemistry was awarded to these 3 gentlemen, Martin Karplus, Michael Levitt, and Ari Rochelle, and for the development of multi-scale models for complex chemical systems. It began with work that Michael Levitt did, in which he classified proteins in 3 different classes. And over the course of this lecture here on Thursday, hopefully you will see these images often enough to sort of understand what you see. Essentially, those are on the left, there are proteins. Those are 3 different protein structures, 3 different proteins. On the left here, the red one with the rods, there are so-called alpha helices. On the middle one, you see one that has the yellow beta strands, and you can imagine them really like fingers. They are aligned with each other, these arrows that you see here. There's a little bit of an alpha helix here. Then there is one that has sort of both. And this is the simplest classification there was. There are some that have the red ones only, some that have the yellow ones only, and some that have a mixture. And you already realize in the world of biology, in the middle are the ones that are all yellow. And it's very easy for you to see, for computer scientists, they are not quite all in yellow. This is what biologists call all yellow. Okay? So this is an exception. Biology is full of exceptions, and you classify systems by thinking that exceptions are exceptions and you keep them out. This classification has been done by Michael Levitt, who then came from physics. Now, essentially, the reason why this is important, these structures, is because the molecular details of the three-dimensional structure of a protein are important for the function. If the protein doesn't function, there's a problem in your body. So, from structure comes function. Now that means if I could predict structure, if I could sort of take the computer and predict how this object looks in three-dimensional structure and how, then I could possibly predict how it functions. Now, in principle, you could do it from first principles, from physical equations, but the system is way too complex. And even for the binding, the functional site, let me just pick out an example here, or an example here where the DNA wraps around something. You can imagine there's this long string of DNA, there's this one protein that does something to it. The part where they actually interface, where they touch, where they connect, is relatively small with respect to these two things, to these two objects, DNA and protein, right? So the binding site is much smaller than the entire protein. So the idea was maybe we can at least do more computing intensive physics where the binding is. And the remainder of the protein we sort of treat as an average. And that essentially is the idea of classical physics versus quantum physics. So quantum physics is in the binding side, around it you try it with classical physics. And that's exactly the idea from 75 that brought the Nobel Prize in 2013. Now, I want to put out two parts here. The first one is a quote from Michael Levitt. It's sort of nice to see that computational science is acknowledged. So he very much sees this as a prize for computational sciences. And in some ways it is. Because when you look at the statues for the Nobel Prize Award, there is in fact for chemistry it's very difficult. So the different fields are different in terms of how they give out Nobel Prizes for theory. Chemistry is very complicated. So it's explicitly written that you need to do something that affects, that has a technical application or has theory is discouraged from Nobel Prizes in chemistry. Nevertheless, they made it. And this is ultimately because their computational work was so essential for a lot of follow-up chemistry. The other point is another important point made here by Ari Rochelle, which is that by using what you get from chemistry and physics and putting it into a computer, you learn something about the protein that otherwise you cannot learn. You cannot observe the protein, okay? You can only see this. You create a reality in the computer that there is no other way to see. It lets you then predict something. It lets you then sort of design a drug. And you see whether the drug works or not. And that sort of tells you whether the model was right. But you only actually see it in the computer. And that is another reason, I believe, why this went to the Nobel Prize. Okay, let's get a little bit slower into the background. Life is diverse, as you know. How does life work and what is common to it? When this image was done by Leonardo da Vinci, the idea of this image is that everything can be described by shapes. So there is a balance between the universe and us. And this balance is described by mathematical shapes, circles, simple shapes shown here. That's the idea of this image, and human is inside of this and is explained by these shapes that he draws around, the regular shapes. Today we're not entirely believing this anymore, but a lot of physics is still built upon these simple principles. Now when we get into biology, the thing is much, much smaller. Inside is the cells, right? And this in fact is what is common to life, or one thing that is common to life are the cells. Is there another thing that is common to life? If I asked you what sort of makes up life, and now let's talk about the sort of thing, not a principle. What's water? That's a great answer. We will get back to that answer. Certainly water is absolutely coming to life. No life without water. But it's also true that there's a lot of stuff that is not life that has water. Let's talk about ice. I love ice, but it's not. I wouldn't call it alive. It makes me possibly alive. Yes, that's another important point. DNA, yes. DNA, protein, RNA. And those are 3 biomolecules. Does anybody here have enough biology background to put them into place, those 3 words? What do they have to do with each other? So they are all these 3 biomolecules. Is it trivial for all you guys how they relate? Who knows clearly, if I were to sort of give you 2 minutes or 3 minutes to sort these words and find relations, is anybody, who would be able to do that? Okay, you do need my introduction. This is not, I hear laughter. I just don't know anymore how much they learn in schools. So you guys come from schools where you never learned DNA, RNA, and protein? Why do they let you in here? Yeah.
I don't know if that's correct, but I think protein gives rise to RNA, and RNA then forms complex structure to give rise to DNA.
That's a great answer. This is the historical answer. And we will get back into that. So, actually, I'm not sure how much we get back into that. So let me just take the moment. How did life start? And this gentleman here gave the answer that is currently assumed to be the case in some sense, in a slightly convoluted way. So it started with RNA, is the current belief. Now RNA could not do everything it had to do to replicate. It found a slave, proteins. They did the enzymatic activity for the RNA. And that bore the protein word. And proteins, sort of, they are the slave unit for the RNA. In some sense, it's like a war. They took over and created another piece that in fact is the information content, and that's DNA. And that ultimately is the story here. DNA is the information. That's how the information is stored. That's the library, right? It's a library where the code is a 4-letter code. RNA is, and that is a complete incorrect oversimplification, an intermediate step. So also the information flow is not quite right. It goes back to both arrows. But ultimately this version is the simplified version and has a lot of truth to it. This is the dominant truth, let's say it that way. DNA stores the information. RNA is an intermediate step today for life, and that creates the protein. The protein is the machinery of life. In terms of the way it's made, again, DNA is 4 different nucleotides, 4 letters. RNA is also 4 letters. Just 1 letter differs between RNA and DNA. Protein is 20 amino acids. How can you get from 4 to 20? If you have a question, just a sec. If you have a question, please ask it. You're not the only one. Please ask it loud, unless it's about the party this evening or something. No? Sorry. So how can you get from 4 to 20?
Combination.
Combination is the right word. Here is the first one.
We take several nucleotides into one amino acid.
Several. Yes. How many do you need? Somebody else. 3. So 2 is 16. 4 times 4 is 16. 2 wouldn't do. 16 times 4 is 64. We have only 20. What do? You have way too much. Okay, 2 would not be enough. 3 is too much. What do?
Either you don't work with some of them, or you use several triplets to encode the same.
So one way could be, he says, that you could use, and he's already calling it the name triplet. Sure, it's a name. There are 3 nucleotides that together form 1 amino acid. So one is, you could just not use them, use only 20, find the 20 that you want, or you could use some more often than others. So you simply could essentially build in redundancy, right? You cannot, in fact, you could have one way of doing it, you could simply have 20, 64 possibilities. So you use every single one with 3 different triplets, right? 3 times 20 is 60, and then you have 4. Actually, those 4 you could use to decide how you start reading and stop reading and things like that. Do you believe nature is done that way? Who believes it's done like that? It makes a lot of sense, right? So nobody believes it's done like that way because you all don't believe in sense. Is that what it is?
It just can't be that simple.
It can't be that simple. I very much, this is an interesting answer. I do disagree with that. It could be that simple. The problem is that there are so many other things that have to be optimized. And the way the 4-letter code is translated to a 20-letter code is a process, and you have to optimize that process. It has to have a certain speed, and you have to optimize that speed. And part of that is actually coded into which triplet codes for which amino acid. So one thing that you can immediately see is that amino acids that are more potent, you will code with more triplets, right? And that is in fact the case. It's not that simple again, but the algorithm is not 3 for everyone, some with more and some with others, and that has all kinds of reasons. Just words, biology is full of words. I do not expect you to remember these words, but I do expect you to remember the idea, and that is going to be true for the entire lecture. For the examen, it will become a little bit more learning by heart than I really like, because again we have 220 people here taking the lecture, and there's a lot of overload to test the understanding of 220 people. So there will be a little bit more, you have to learn certain things. But it's not about words, it's principles that I want you to learn. So transcription is going from DNA to RNA, translation is going from RNA to protein. When we look at the world of 3-dimensional structures of protein, you see there are different scales here. Those are all different size or different proteins of different sizes. Some are very, very, very big. Okay? And that brings me to the next part of images. In this slide here, what is similar? Carbon. Is all carbon? Oh, 2 people shouted carbon. Carbon is similar to carbon. Is that all carbon? Is that what it meant? I take it that this is what it meant. They're all crystals. This I totally like that idea. All 4 things are similar. Yes. So you meant these 2? Yeah, well, sorry. Yes, that's a very good question. What do I mean by similarity? So, okay, so we are in a part of the lecture where we talk about guessing. Science has to do sometimes with guessing and then finding the right scientific answer. Why what you guessed may be right. You have to start with a hypothesis. And this hypothesis should be as scientific as it can, but sometimes it's an intuition. Science is about really nailing it or seeing whether it's right or wrong. Okay? That's the scientific process. So I'm flashing a lot of pictures that you will not be able to clearly answer, and where I cannot quite give you the definition. Later the definition becomes clear, and here's one of those things. In this particular case here, we have 2 objects that are silicon, made of silicon. So that would be one way of defining similar. They use the same material, all right? And again, in the sense of crystal, they are sort of semi-crystal. The glass is almost like a crystal. It's fluid, as you know, as some of you may know. Glass is flow. They're not quite crystal-like. The packing is slightly different. But here, the similarity is really the material. This is graphite, diamond, and again, it's a similar story. There's diamond lattice underneath. There are different reasons to call things different, with things similar. So typically, again, in chemistry, the way we do that, we have atoms and we have a periodic table, and the periodic table is built up by some principles. Now, if I asked you what's the molecule that dominates our body, and I believe the answer already was here before, water. True. How much?
70%.
At some point, I really appreciate your speed and rightness, but at some point I'm going to exclude you because you're always right on. Yes, it's about 70 or a little bit less than 70%. Now, if I asked you what other than water is most important in our body? Carbon, yes? Anything else? Yeah, well, now we're getting into the issue, if we call water the most important, then oxygen clearly is in there, and hydrogen and oxygen is it, and carbon is number 3. So the first 3 were all named in this group. And that is amazing, but I still want to point out the numbers here. The next slide was missing. So you clearly all essentially covered most of the pie. Most of the stuff in front of you is up there. And then comes nitrogen, calcium, bones, phosphor, and those are the big ones, okay? And then we get to the small ones. All of this combined, this is 10, combined is 96.5% of our body weight. The entire periodic table, all the types of enzymes that you have, vitamins that you have to eat, all the diversity of food. In terms of body weight, we are pretty simple. On that sense, what is this? Oh. No, somebody said it, right? Did I hear that right? Yeah. But it was somebody else. It's E. coli. Gut bacterium. Now, wait a minute. This is twice. What is this? A virus. This is really perfect. This is not, this particular case is not quite right on, but it looks like it. This is a different way of looking at E. coli. It's a different resolution of E. coli, in fact. And this is the reason why I show you these images. And I'm going to have a theme of a couple of going images, because ultimately in this lecture we talk about three-dimensional structures of proteins. And we have an image of what a three-dimensional structure is. We cannot look at them like that. The images that we build are all examples, are all ways of visualization. And that's what this lecture is supposed to bring home. That images imprint ways to think about it. They are not the truth. What is this? And salmonella, this is the same here. And here we are to the avian bird virus that I heard before. This? Guys, have you heard the lecture before? Or what? How do you know this? Okay. This is HIV, yes. Now, I have to challenge you. I had to challenge you. I'm sorry. I really appreciate anybody jumping forward. Any idea? Where did that come from? This is the first. It's the third time I give this lecture. There were 600 people in front of me. This one never anybody got. They got E. coli, they got all kinds of things, but Velcro. How can you know Velcro? Yes, it looks like it, sure. Yeah, you know. Every single time I open my shoe, I feel this is exactly the way it looks. Interesting story, however, it comes from a guy, Georges Mestral de Mestral, who walked his dog. And when he walked his dog, he saw the burr in the fur of his dog, and the burrs just sticking to the fur of the dog because of the same principle that he put into Velcro. By the way, he never patented it. But he pushed this whole thing through, invented it, and made it to the industrial design. Bacteria differ in shape, in the environment, in the way they live. Some of them live alone, some of them live in colonies. There are even sort of multicellular type of colonies almost. What they have in common is ultimately that all bacteria are essentially beings that are single-celled. Okay? There's only one cell. Even if they live in a group, there's a single cell, and this single cell essentially has a wall around it, and it has something inside that does all the processes of life. Again, the entire apparatus that is working inside are proteins, is made up of proteins. The eukaryotic cell is one, most eukaryotic cells live in colonies. We are examples for that. Yeast is a single-celled eukaryote. Baker's yeast is the one that you use for beer. But they all have in common that the structure is more complicated. So there is the cell wall, but there is a structure inside. There are several compartments, and these compartments have a special function. And that again helps to sort of increase the complexity in the way we can operate. Do we carry bacteria? Well, yeah, sure, we carry bacteria when we are sick, right? But do we carry bacteria when we're not sick? How much?
I'm not exactly sure if this is true, but I read before that there are more bacteria cells in our body than bacteria cells.
So there are more bacterial cells in the body than eukaryotic cells. This is true. In fact, it's even worse. In some sense worse. There's more bacterial DNA in front of you. Here stands more bacterial DNA than eukaryotic or human DNA. If you ground me up, most of what you found, I don't suggest you do that, but most of that really is bacterial DNA. Just because bacterial cells are relatively small, there are a lot of them. The DNA is less DNA than the human DNA, but the eukaryotic cells are so big that ultimately in terms of weight, there have been the numbers out there that we carry 2 kilo of bacteria in us. Or somebody such as my weight, I carry roughly 2 kilo of bacteria. This may be an overstatement, but it's certainly not far from the truth. And it's certainly true that a lot of these bacteria are absolutely essential for me to feel good. And they are not only in one part of my body, they are throughout. They are in the skin, they're in the eyes, they're in my guts, they're in my teeth, they are throughout. And the colonies or the groups of bacteria constantly changes. Depending on how I feel, depending on how I feed, on a variety of things, and this is a big field that is still growing. Now again, what is common is DNA. Where do we have our genes? Do you know that? So the genes are essentially wrapped up in structures that are called chromosomes, but all of them in eukaryotes, in fact, are in the nucleus or in the mitochondria. Mitochondria is also often considered the sort of power factory of the cell, but the nucleus is really, in some sense, you can think about it here in green. The green is the actin here, the red is mitochondria, blue is nucleus. So you see the core here. I'm sorry, I would have to make it a little bit darker, much better on my screen. Maybe you can sort of guess that there is a blue here. That is the nucleus. Mitochondria are sitting around it in red, and green are the actin fibers. Muscle fibers are also actin fibers. You can think about this as a system of tracks that runs through the cell. So most of the exchange is done on these green tracks. Okay? Unlike train tracks, these are active tracks. So it's not that the train runs with a big diesel engine or something like that. In this particular case here, the track has ATP in it or has some device in it that makes the train move on step by step. Okay? So the track pushes the train is the system here. But again, most of the export in the cell is done on these green tracks. DNA is in the blue, in the red. Here again in the background of the protein, and here in terms of the machinery of life, the information flow. What is the smallest building block of life that can replicate? Protein, quarks, cells, organelles. different cells in typical human. How many, so I'm not asking, I have 65 trillion cells, okay? But what I'm asking here is how many different types of cells do we typically have, all of us? 20, 200, 400, 1,000? Nobody believes in 20. But it actually is remarkably similar. So it's somewhere between 200 and 400. So the number 200 is closer to the truth than the number 400. What are the parts of the cells called? Cellular parts, organelles, organs, cellular plus. So sometimes life is unfair. Yes, very good. Which part of the cell is called the powerhouse? Should I stop the introduction? Great. There are two different ways of saying that. One is that it's not necessary what I do. Another one is it's great to have a group who knows so much. What is mitosis? What part of the plant cell is involved in photosynthesis? Who first used the term cell? I would have taken Captain Hook, it's much nicer. Anyway, how many elements are found in the This is the first time. Again, I've given this lecture before, this speed I've never seen. How many elements are found in amounts larger than trace amounts? I already have in our bodies 11. I already gave you the 11. When communities of living things interact with non-living things, they are called population, community, biosphere, ecosystem. The most common molecule in the body is blah, blah, blah. Okay, now, can you define life? I take it, so let me just, for the ones sitting in the back, there are a few people in the front here who vividly shake their head, indicating that there is no definition of life. This is the way I read that, and that is correct. Which again is where I come to biology. The way we typically describe in biology is by sort of listing a set of features. And features, can you name some features? Can some people in the room name some features that we commonly associate with life?
Your ability to reproduce.
Reproduction, yes. Metabolism. Is it louder? Breathing. Energy exchange, yes.
Response to stimuli.
Yes, response to stimuli. Adaptation, that's an important, very, very important one. And let me just, you guys really should be impressed by yourself, how many you picked of these. But that gets to the next one. Oops, my battery's running out. Are viruses life? I hear yes. I hear maybe. I hear sometimes. That's an interesting one. Sometimes I like to be explained. Can whoever, if you want to speak up, who said sometimes? Yeah.
If it's not in contact with any biological organism, then it's not alive. But if it's in contact with some biological organism, then it's alive.
Yes, that's a very interesting definition. I've never heard that one. So he says viruses are life if they are inside of another organism. They are not life if they're all by themselves. And that is absolutely is a wonderful way of putting it. Because what we expect from these definitions of life is that whatever I call life has all those features. And viruses don't. They need a host cell to reproduce. All right? So this definition is totally right. As soon as I have another host cell, they can reproduce, and they will actually fulfill all of these criteria. It's an interesting point. Now, things got a little bit more complicated over the last 3 years because people have discovered very big viruses. So let me define very big. So HIV has 5 genes or 7 genes. Most viruses are in the sub 10 genes category. Some viruses have 20. There are even viruses known that have 30 or 40 genes. That sort of is, a typical bacterium has between 500 and 4,500 genes, okay? Now they have discovered viruses that have 3,000 genes. They have discovered viruses that have actually membrane around them. Have viruses that essentially have all the types of features that you would call life. Why do you call them viruses? Well, there's a biochemical probe that distinguishes between viruses and bacteria, and by that probe, they look like viruses. So this is something that depends, and so something is changing here. Again, the central dogma as an information flow is that DNA, the flow of the information goes to RNA, goes to protein, and then there are loops in there. Again, also, this leads to the sort of central dogma of it is more related to the proteins, where this DNA sequence, the amino acid sequence, leads to an amino acid sequence. The amino acid sequence you can perceive or think about as a stretched out chain, a pearl chain, where you have 20 different colors, 20 different amino acids. And what you can do when you string up the pearl chain, you use different colors. You can use 3 reds in a row, you can have red, blue, green, red, blue, green, red, blue, green. You can create patterns, you can do whatever you want, okay? And all of these mixtures are different proteins. A particular mixture leads to a particular 3-dimensional structure, okay? This one mixture essentially always gives the same structure, or it doesn't survive. Because the 3-dimensional structure is relevant for its function. If it doesn't do the function in a reproducible way, meaning the same sequence always leads to the same function, then something goes wrong, something is broken in this sort of information flow. And when something is broken, this thing will not survive and we will not see. So for all the ones we have seen, they are exactly that way. Sequence leads to structure, leads to function. Okay? Again, I talked about the triplet, the DNA triplet, codon. So inside is the first codon, the second pi is the second codon, and the largest pi is the last codon. And there you see the different amino acids, the 20 different amino acids here all at the end. And there, for instance, the T is coded by ACC or by ACA. ACT is all leading to a T. And you immediately see that some amino acids, some letters here have a higher variety of triplets than others. Okay? And that we get to...
When the RNA copy is complete, it snakes out into the outer part of the cell. Then, in dazzling display choreography, all the components of a molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. Each transfer molecule carries a 3-letter code that is matched with the RNA in the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecules. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second. And as a result, our muscles, brain, and all the vital organs in our body receive the oxygen they need.
Okay, so the hemoglobin is in red here because typically the color of blood is red. That comes from the oxygen. Hemoglobin is also the first protein for which we actually could, for not we, but for which the three-dimensional structure was determined by Perutz, 1960. How do you determine a three-dimensional structure of a protein? Any idea? So you cannot look at it. The dimension, there are two things. The size and the dynamic situation of a protein is such that you cannot see it under the microscope. So the only way you can actually see it is by freezing it in a crystal and then pulling beams onto the crystal by diffraction studies, by X-ray beams. And that's the way, from the scattering of the X-ray, you essentially see what atom is where, and from that you can construct the three-dimensional structure. As you can already sort of guess, the complexity of an object with many thousand atoms, it scatters X-ray beams, and you have to reconstruct the three-dimensional image. That is not that easy, and it takes a long time. And in fact, to get the protein to forming a crystal is also not that easy. Now, I said that bacteria essentially have an outer membrane, and inside there's all the machinery of life. Okay, so I imagine this like a sort of plastic bag with a liquid inside, and inside swim the proteins. Is that the way you imagine a cell? If anybody feels that this is not the way they imagine a cell, please tell me how you imagine a cell. If you feel you don't imagine a cell, start imagining one now. Find your image. Anybody? How do you imagine a cell? Is this water? Okay, it's not a plastic bag. It's a lipid bag. Yeah?
Probably kind of like this, but there also must be some openings where you can...
Yes, that is a very, very important point. I left something else. We will come back. It's going to be a central part of this, later part of this lecture. He says there must be a gate, there must be an opening. Just really, it's true. Imagine the cell wall like in a medieval town, a wall around it, and then you must have a gate because there's a constant change of exchange of energy. Okay, breathing is one thing you said. Now. Other than that, so again, I have the wall, imagine it like a stone wall, it is a lipid in reality. And inside, if you imagine it like a medieval town, then you have these streets, ways in there in which you have sort of a relatively free flow of people. Free flow of information. And this may be sort of an analogy to the idea that I had a plastic bag with liquid in it. Both of these images are totally wrong. Okay? And I'm unfortunately not at the right place in my lecture to say what I say now, because I don't have the image that I wanted to show you for that. But the way to think about a cell is a watch. The density of a cell is the density of a solid state. It's not a liquid. The cell inside, I showed you the actin tracks. Yes, those are actin tracks, but actin tracks in something that has a density like a solid state. Okay, so there's an immense amount of movement in a cell, but this immense amount of movement. Imagine it. I don't know. Have you been on Fifth Avenue on Christmas in New York? So it's densely packed. Okay, and then there's constant movement in all kinds of directions, and not in that direction. On a 2D level on the street, everybody passes everybody, and it's amazing how that happens. And this is the cell. Okay, clock, watch, cogs touching each other, and those cogs are the proteins here. Everything works together. Now let's get back to numbers here, and numbers begin in the world of biology with DNA sequencing these days. So you essentially sequence a piece of DNA by getting a rapid image of, in this particular case here, fluorescent light, where every base, so each letter of DNA emits a slightly different light pulse and that you can measure, and then you can essentially see what is in this DNA. With that, and that's by now outdated here, this is early 2012, a machine called MySeq could run in 6 hours. In 6 hours it would produce something in the ballpark of 10 terabytes, okay, per day. 10 terabytes. So you have a little machine. Now this thing here is the size of a printer. Now imagine you have a printer in your office and that printer produces 20 terabytes. This is 2012. Okay, it's even difficult today. You couldn't do that. And you're going to carry all these hard disks in and out of your office. Then there are these bigger things, HiSeq Illumina. They do 24 gig per day, and again it's outdated. Then there is a group here, BGI, the Beijing Genome Institute in Shenzhen, that has the largest collection of sequencing machines worldwide. In fact, the BGI has about half the facility of the entire planet on sequencing in that space in Shenzhen. Today, these things are even bigger, and with these things you can do a lot of sequencing. And for instance, you can sequence entire organisms. The first that was ever sequenced was in 1995, Haemophilus influenza, it's a small bacterium. Several others followed. What I show here is the genome size. So this is in fact the smallest bacterium, Mycoplasma genitalium, 470 genes. Then that was the first one that was sequenced, 1,700 genes. And we get into other ones. E. coli, I showed you before, 4,000 roughly. Here we have the first eukaryote, yeast, 6,600. Fruit fly, worm, this is a weed here, rice, and Homo sapiens. Now, Homo sapiens here is an estimate from 1999. In fact, 1999, just briefly before the human genome sequencing was done, it was supposed to be finished in January 2000. Did not quite happen. But briefly before this January 2000, there was this idea that humans essentially have 100,000 genes. And that was done after 5 years of sequencing, just before, so the project runs for 5 years, you have three months before completion, or less than 3 months before completion, going to the press, and you believe it's 100,000 genes. Then there's this meeting at Cambridge, at the Sanger Institute, in which they said, well, it's not quite 100,000. We have a tiny correction here. The number is, oh, sorry for the animation. The number is actually 30,000. That's biology for you, okay? You work for 5 years on a project, and you're almost there, and suddenly realize it's not 100,000, it's 30,000. This is the amount of discovery that you have in biology. And let me put it into a background here of physics, speed of light, and the novelty in biology, the way I call it here. And so on the left you see the degree to which we gain information from 1999 to 2015, because 2015, now we know it's 20,000. Or we're pretty sure it's about 20,000. So this is the speed of light in 1690, okay? This is the speed of light today. And look at the gain of novelty in physics over time, and look at the gain of novelty in biology. This is provocative, but there's a lot of truth underneath. The amount of novelty that you create in biology is just stunning. Now we know it all, right? We have the entire human DNA, we have the entire genome. And Anna Tramontaro said, like, for every good manual, either you never find what you're looking for, or if you actually find it, it doesn't help you because you don't understand it. And you know, the analogy here is this image, you take the car apart, or the 380, you take the plane apart in all its parts, and somebody tells you put them together. And that you have to do without actually having any manual or anything that says how they come together. Nobody ever showed you how the cogs work, right? And that's the situation in biology. So we have the parts, but already on the slide before here, you see that in 2000 we had the parts. About 30,000 parts. From 2000 to 2015, we moved from 30,000 to 20,000, so we reduced the number of parts. So even knowing what a part is is not that easy. Even that, in many cases, took 15 years. Having them in the tube still hasn't happened. So for most human proteins, we do not know what they do. In fact, we have no idea what they do. Okay? For some fraction of the human proteins, we have a good idea what they do. And that fraction is relatively small. And that is not because there's not a lot of money going into this project. It is because it's very, very complex. That gets us back to the central dogma again. Sequence leads to structure, leads to function. And in fact, Christian Anfinsen is the one who got the Nobel Prize for actually showing that sequence leads to structure. The same sequence always leads to the same three-dimensional structure. If that is the case, then we should be able to predict protein structure. There is another example here, or another step. I said RNA is the intermediate step. Now, an intermediate step, in terms of information control, means it's not really important, right? A few years ago, there was another publication that overnight essentially said that there is an equal number of RNA molecules that are actually doing something that we don't know about as the entire protein machinery. So there's a huge universe that we haven't seen before. Still, the central dogma holds, and we want to predict from sequence protein structure. What are proteins? Now, proteins are doing defense. They're antibodies, they are structural proteins, they're collagens, they're muscle fibers, they're enzymes, they do communication, they bind ligands, they transport stuff through the cell, and they store, for instance, oxygen or iron. I showed you some of the gallery already before in a brief moment here. So this is a set of membrane proteins up there here binding in the lipid. Proteins come in different sizes and different functions, and they're even bigger, multi-chain or things that are put together. And so this is the cover of a rhinovirus, which again is a huge protein. That's one way of thinking about them. That's another way of thinking about them, is a long sequence. How many do we have in human? Again, I already said that is 20, at the moment we believe 20,000. How long are they? So how many of these letters do you believe you have? Well, ultimately the answer is somewhere here. They're between 30 and 30,000. And this again is an immense scale. Just think about this. 3 orders of magnitude. Just, a protein can be 30 residues long, it can be 30,000 residues long. Okay, 30,000 is extreme, 30 is not, the 30 residue is not that extreme. 30,000 is extreme, that's one. The median is around 400. But when you look at a number like that, to just even quote a median doesn't really make sense, right? It sort of falls short of convening the information. That means, ultimately, you know the answer to this question. Do they consist of units? Yes, they must. There's absolutely no way that you sort of string them up in a way where the same thing has 30 and the same thing has 30,000. There must be some repetition of something. And that something is typically called the domains. Now, how many proteins are known? What do you believe? 20,000 clearly is a lower estimate, right? We know, I said by now we know 20,000 human proteins. So how many proteins are known? Sequences. What do you believe? I heard a number, just a little louder. 100,000. Yeah, it's a little bit more. So 100,000 would be, we know of 5 different types like human. So human, mouse, rat, fly, worm, we have already 100,000. It's a little bit bigger. It's at this point in the ballpark of 85 million. Now, on the number of 85 million, so this is the growth over time of the protein sequence database, and you see a dip here. And the dip ultimately had to do with the point when there is a new sequence submitted, do I take it? Well, if it's already in, then I don't take it. But what if it is a shorter fragment of something that is already in? Do I still take it? And under that point, essentially, they said, well, if it is a shorter fragment or something longer, so if it's not 100% contained in the database already, is a new entry. And at that point, they decided that everything that is 100% identical, they will sort of merge into one mega entry. That's why it dropped. Again, it's not up to date here, by now it's above that number again. or just about that part. It's about 98 at the moment. Now, here's another reality that is important for anybody in this field. So the way I show it here is the way when our group came to Germany. So when we came, it was about 8 million in 2009. In 2014, it was 55. So this is sevenfold in 55 months. It's roughly double every two years, right? It's actually faster than computers grow today. But similar ballpark, right? Now, the problem is, so how do I learn about a protein? I take the sequence, and my first question is there any other sequence or a protein that is similar to it in the database? Just a sec. So in order to find out, I have to take this sequence and compare it to all the in 8 million that are there, or 55 million that are there, right? And that, unfortunately, sort of leads to a situation where the number of comparisons grows by 47-fold in 5 to 57 months. So this is 20-fold increase over 2 years. And that is much faster than computers. And the only way to do that is by computers. So how do we do this? Well, we're trying to get more advanced algorithms. We are trying to have shortcuts, is ultimately the answer. But when I started in this field, the reality was that every 2 years things were easier for me, and now the opposite has happened. It's getting more and more difficult. Here's by the way, the last image, the amount of space we need to do some of the storage that we have in this time frame. And again, it was much easier when we came with 5 terabyte in 2009 than to juggle in 2014 300 terabyte. So 300 terabyte today is really not trivial. Just write it onto a tape and see what happens. Or do a backup of it, and you'll see how non-trivial that is, even today. Now, the other issue is that I talked about 85 million is the number of proteins now. When we look at the number of proteins with annotations means I know something about what it does. Okay? I know more than just the sequence, just the amino acids. And you see that the scale here is 600K. So this is more the ballpark that you said, 100,000, right? It grows, so somehow here it sort of tethers off a little bit because people spend more time on doing more detail. The database here, shown in red, or the increase shown here, is called SwissProt, and now what you see here is the SwissProt annotations versus all sequences in red. And you see that, in fact, the annotations lag behind. When we ask, how is that now for proteins of known three-dimensional structures, the crystals that I talked about? So all the details in terms of three-dimensional structure, the answer is here. The protein database, proteins of experimentally known three-dimensional structure, again, through crystallization, through X-ray, and then the mass structure determination. I will briefly get back to that in a moment later. We have at this point about 120,000. And when you look again in relation, so that's a logarithmic scale, so that's the SwissProt number here, PDB, and the UniProt. So by far we know more sequences than we know structures, than we know about function. The gap in between the red and the green, or the red and the blue, that is what we're trying to fill in with prediction. That's sort of the subject of this lecture here. So for the gap, what can we do in order to sort of have some annotations, in order to have some ideas about structure, about function? Again, proteins are the machinery of life, out of the genetic code. Now, one thing that I can ask is when I look at different kingdoms, so I look at eukaryotes, bacteria, and archaebacteria, so different types on the tree of life, and I ask, what is their usage of amino acids? So when you look at the entire sequence of the entire genome, or all the proteins, do they use the same amino acids? What do you believe? Bacteria and eukaryotes. They do, because they're all life, you say. Is that what I understand in the way you wave? Of course they do. For many people this was a big surprise. Yes, so the height of the letter here is the 20 amino acids. The height is proportional to the usage of that letter in that organism. You already also see that some amino acids, the H here and the C, are much less used than others. The A is much higher, the L is much higher, the V is much higher. So you see that they are used differently, but you also see that they essentially use the same between the kingdoms. Now this is surprising because we know that the so-called codon usage, so which triplets are used to code for which amino acids, and here is the other reason why this is more complicated, I don't remember who said it cannot be that trivial, is that different organisms in fact use different triplets, and they use different triplets because they get, essentially the DNA is in different environments. And the susceptibility of different triplets to environment is also very different. So there's yet a different reason why you have different triplets, the generation of the code. And you can use those triplets to in fact predict the organism from the triplet usage. So it's actually surprising that at the end of the day, on the level of the amino acids, we have a sort of equal view again. Okay, the